It's obvious from the close connection between people and their pets that quite a bit of communication goes on in these relationships. But the true extent of this communication is unclear. For example, there's an ongoing debate as to whether dogs can actually understand the words that people are saying, or if they're just picking up on nonverbal cues, such as tone of voice or body language. In a recent science paper published in the Journal of Neuroimage, a group of Hungarian researchers developed a set of experiments to try to get to the bottom of this. Specifically, their experiments were aimed at answering two questions. Number one, can dogs differentiate between human language and scrambled or gibberish human language that has the same tonal qualities? And number two, could dogs differentiate between a human language that they were familiar with and a human language they had never heard before? Answering these two questions should give researchers more insight into the extent to which dogs can actually perceive and understand human language. So, how did the researchers attempt to answer these questions? The basic idea is that they plan to expose dogs to human language clips from different languages and observe their brains using a technique called FMRI, which I'll talk about in a little bit more detail in a minute, to observe differences in how the languages stimulated their brains. So they got 18 adult family dogs. All had been previously trained to be able to sit still in an MRI machine. 16 of the dogs had been raised in Hungarian-speaking homes, and 2 of the dogs had been raised in Spanish-speaking homes. Linguistic material was then developed, which consisted of recordings of the 21st chapter of the book The Little Prince, read by 2 different native female speakers who had similar voices, one for each language. Then, they made scrambled or quilted versions of the Hungarian and Spanish language clips to serve as controls. The idea is that they would have the same average frequency and tone, but not have real human sentences and words because they had been scrambled or quilted. The dogs were then exposed to the different clips while their brains were being observed via fMRI. Now, before we get into the results, I'd like to take just a minute to explain what fMRI is and how it works. So fMRI stands for Functional Magnetic Resonance Imaging, and it relies on the fact that hydrogen atoms in your brain can actually be stimulated by a strong magnetic field. The hydrogen nuclei can absorb energy from the magnetic field and then create a brief, faint signal that's called an MR signal. This allows mapping of the brain structure. What's interesting is that the strength of this signal actually depends on what's going on in the brain at the time of observation. So for instance, oxygen-rich blood versus oxygen-poor blood have different magnetic properties, which lead them to having a different level of signal depending on the amount of blood flow and blood oxygenation in the brain. As it turns out, neural activity in a specific part of the brain can cause a big change in blood flow, which is significant enough to actually lead to a change in the MR signal while there's a specific activity going on. For example, if you sequentially tap each finger of one hand against the thumb as fast as you can, the blood flow in the motor region of your brain increases by about 60%. Similar changes in blood flow can be stimulated by visual cues, by looking at a certain thing or closing your eyes. For example, the simplest fMRI experiment, a subject will alternate between periods of doing a particular task and a control state. For example, in this study, you can see right here, this graph is 30-second blocks looking at visual stimuli compared to 30-second blocks with eyes closed. And you can see that the open and closed states, you're seeing very specific changes in the neural blood flow. Overall, this means that fMRI can actually be used to create correlations between specific stimuli and activities and activation in specific parts of the brain. And this is really going to be the basis of the data that we're going to look at in this study. Okay, so let's get back to our dogs and MRI machines and see what we can learn from this data. Specifically, what we're looking at is maps of the dog brains with 2 different states compared. So first, we're going to look at the orange, which is basically showing the difference in brain activation regions for natural speech versus dogs exposed to the scrambled speech that we talked about. Specifically, the blood flow, which remember that's a proxy for brain activity, was significantly different when the dogs were exposed to natural versus scrambled speech. Moreover, these differences were found to take place in the auditory cortex, which is the part of the brain that has been identified to be sensitive to sound in many past studies. So to try to answer our first question, which was, Can dogs differentiate between human language and gibberish with the same tonal qualities? The answer appears to be a big maybe. There's definitely a difference in brain stimulation, but we aren't sure if this is because dogs have a special relationship to human language as such, or if it's simply because they have a special recognition or relationship to unnatural sounds, such as a scrambled voice recording. That said, it does seem that the researchers took the time to down-select voice recordings that were rated as more natural compared to some of the others. So I lean towards the former explanation, that the differences in brain stimulation has more to do with responding to human language rather than just responding to a strange or unnatural sound, but we really can't be sure, so that's why I'm kind of rating the answer to this question as maybe. Okay, so looking again at figure 2, you can also see from the blue highlights that there was a significant change in blood flow to specific regions of the brain for natural speech in a familiar language, Hungarian for most of the dogs, and an unfamiliar language, which was Spanish. Also, the researchers found it interesting that it's a different set of locations in the brain than for the differences between natural and scrambled language, which basically argues that there are two different processes going on for these two different sets of differentiation. Specifically, the regions of the brain that were highlighted in this second experiment have in past studies shown sensitivity to things like pitch, emotion, and voice identity. Also, these regions have shown sensitivity to exposure to trained words versus untrained words, which may indicate that these parts of the brain can help the dog detect novel words versus familiar words. So in terms of answering the question, the second question, can dogs tell the difference between two human languages? The data actually indicate a tentative yes. So the researchers believe that these differences in brain activity, coupled with what we know about these brain regions from prior studies, support the idea that dogs, during their lifetimes, learn the regular patterns of their home language to a degree that they can actually recognize and differentiate it from an unfamiliar language, even when clips have been selected to have similar rhythms. That said, something I really appreciate about this study is that they spent a significant amount of time listing some limitations of the study that they hope to rectify in the future. So overall, the answers to the opening questions are maybe and a qualified yes, pending more studies. So in terms of answering our overarching question, which is the extent to which dogs can actually understand humans, this study is maybe a little bit underwhelming. But I would argue that this underscores the fact that science is hard, and if we have this much trouble with a relatively simple question, it goes to show how important replication and rigor really are. The truth is that a lot of the scientific studies that you see, especially the ones that show up in the headlines, are actually, by and large, a lot more tentative than you might be led to believe based on the headlines themselves. On the one hand, I think it's really important not to be too easily led by these headlines and kind of take it at face value. But on the other hand, I think a mistake would be to maybe just dismiss the scientific process overall. So if you're curious as to just how challenging replication and rigor can be, check out my recent video on the replication crisis in cancer research, but only if you're in the mood to be terrified. Well, if you got this far, I want to thank you so much for watching, and I hope you have an awesome day. Bye.